Welcome everyone to the last OTS webinar of 2025. We have an exciting lineup for 2026 already that's starting to be advertised on the website, so make sure you check in and see what's coming up. But we have an incredible way to end the year here with Bruce Sullenger and Telmo Llanga, and they're going to talk to us about blade-directed evolution of SaCas9 gRNAs, how it improves gene editing efficiency. So I'll let the two of you take it away.
Okay, I'll kick us off, Aaron. So it's a real honor and privilege to be invited to present at the OTS webinar. Go back one slide, Telmo. Just wanted to as a quick disclosure, just point out that Telmo and I are inventors on sort of this blade selection technology you're going to hear about today. And also, if people are interested, Duke has filed patents on it, and we're actively looking for collaborators or potential licensees, et cetera. So please reach out to Telmo or myself via LinkedIn, if you have interest, or also if you're just interested in collaborating on this work as well. We'd welcome collaborations. You can also email me at just bruce.sullenger@duke.edu. And I think Telmo probably has the same email too, telmo.llanga@duke.edu. So please reach out if you're interested in learning more about this technology, potentially collaborating, potentially licensing, because we'd love to work with whoever out there and get this approach more broadly looked at and interrogated. So, okay, the next slide. So it's a real honor to be invited to talk about our paper, which just was came out in the most recent issue in Nucleic Acid Therapeutics on BLADE, which stands for Binding and Ligand Activated Driven Enrichment, Directed Evolution of SaCas9 guide RNAs for improving gene editing efficiency. And I'd encourage people to consider submitting things to Nucleic Acid Therapeutics. One of the reasons, other than it being a close alignment with OTS and being the OTS journal, one of the things that was nice that we were very fortunate about is actually the paper was chosen to be featured on the cover of the journal this issue. And as you can see in the covers of NAT are, beautiful and nice way to highlight sort of your work. And the cover nicely summarized, I think, what going to talk today. I'm going to let Telmo do the main presentation of the work that we published. Um, Telmo was a grad student in my laboratory here at Duke. He actually defended in about 6 months ago, and now is doing a postdoc in a very complementary technology-based lab of trying to evolve the protein components of CRISPR-Cas. But in the what he did in my work in my lab and work with us is really trying to evolve the guide RNA. I would say, as many of you know, our group really historically has worked on aptamers and evolving novel RNA ligands or aptamers to different things. And one of the things we got interested in was, could we utilize that same combinatorial chemistry, molecular evolution type of approaches to interrogate and potentially improve the guide RNA, particularly the so-called scaffold or the protein binding part of the guide RNA, which I call aptameric because it binds a protein just like aptamers do. So we really utilized our expertise in molecular evolution and combinatorial chemistry and high throughput screening, as you'll hear, to try to understand the guide RNA for SA, Cas9's guide RNA in this particular case, and think about how we could utilize that information uh, to potentially improve editing, particularly for hard sites, as you'll hear about from Telmo. So I don't want to steal his thunder, so I'm going to hand it over to Telmo to really tell you the story. But again, Telmo did an excellent job. This was a main thrust of his PhD thesis, and you can see it was really well received here at Duke, and an interesting and I think important story. So Telmo, take it away.
All right. So yeah, today I will be going over just a brief introduction to some aspects of CRISPR gene editing relevant to our project, talk about how blade-directed evolution was employed to obtain varying guide RNA scaffolds, and then also speak about ongoing effort of using a one-round blade guide RNA evolution to direct the high-throughput guide RNA activity rescue of scaffolds, as well as future directions in the work that I'm currently doing in the Chory lab, which is a phage-assisted directed evolution, how we're trying to kind of bring things together. So with gene editing, genetic disease is very widespread and affects approximately 17% of the population. Gene editing nucleases can theoretically target any genomic location, but the development costs of it are currently highly expensive. And gene editing on-target efficiency as well as off-target effects have precluded the targeting of a lot of the sequence space. While there is increased access using mutated proteins and also chemically modified guide RNAs, there's still a lot of sequence space that is not currently targeted. Inpatient clinical trials in 2025 for inborn liver diseases in neonates have shown promise, and this leads to ongoing investigation of future clinical trial pipelines going. So gene editing is currently, for the first time, being used also in this case of inborn liver disease in 2025 in neonates. So this is quite a move forward for gene editing. So one of the things that preclude the targeting of an entire sequence space is that a large proportion of CRISPR gene editing is inefficient. In a study by Corsi and Gorodkin et al., the performance of a lentivirus screen was done using surrogate targets that were integrated with the lentivirus. And this allowed this group to evaluate 10,549 sites, where they found that gene editing efficiency in over 2/3 of these sites was not reaching even 50%, which we can see here in this graph. So this is quite a limitation that highlights the need for increasing the proportion of gene editing efficiency. And guide RNA misfolding is a leading cause of inefficiency, as well, normally the guide RNA complexes with the Cas9 protein and forms a ribonuclear protein complex, which is then able to recognize the DNA target. Sometimes misfolded guide RNA between where the targeting region and scaffold regions have base pairing interactions can cause us to produce defective RNPs that have impaired function and sometimes lack binding altogether to the protein, to the DNA target. And current approaches to overcome low target efficiency don't explore the guide RNA region with high enough throughput. While Cas protein mutagenesis has opened up a lot of new targets through PAM sequence modification, and guide RNA chemical modification allows for more stable guide RNAs. Guide RNA scaffold mutagenesis at the primary sequence level has not been explored in extremely high throughput. And this is where SelectS directed evolution comes in, an expertise of the Sullenger lab to evolve aptamers or RNA molecules that have a functional output was applied to the guide RNA of the CRISPR-Cas complex. So in a study that was going on when I joined the lab, Streptococcus pyogenes, SelectS, was employed to evolve the guide RNA of the SP-Cas9, which is the main Cas9 that has been used throughout a lot of studies. And in this study, Bush et al. Evaluated a large library size, and after several rounds of directed evolution, he found that there was 112 in vitro wild-type light cleaving efficiency guide RNAs that out of these, when they were evaluated for cellular activity, 32 displayed cellular activity. And here in the bottom of this slide, you can see that these different targets going in the y-axis combined with the different guide RNAs scaffolds, which are going on the x-axis, produce different levels of knockout efficiency. So here we can see that the wild type, which is the very first one, when it's combined with targets such as target 1, produce higher knockout efficiency, but this is variable. However, when we explore all of the different possible guide RNAs, some of them actually improve the activity of wild type scaffold at different targets, like target 2, for example. So this suggested that guide RNAs could rescue the activity at different targets that were inefficient using the wild type scaffold. So we moved on to explore SaCas9, for which its small size allows for efficient delivery using both AAV vectors and also higher MOI within lipid nanoparticles. Currently, there's a lot of effort into engineering Cas proteins themselves, and these have yielded several new types of Staphylococcus aureus with different PAM specificities. Furthermore, the PAM site of Staphylococcus aureus, which has several more sites of contact with the DNA, can be used for future evolution in things like phage-assisted continuous evolution to create higher specificity using this even smaller Cas9 variant. So something to appreciate here is that both these have a guide RNA that is both 100 bases long and is highly structured. So this made it an ideal candidate to expand our work using guide RNA directed evolution. So now I'm going to go into how we actually obtained and performed this directed evolution. So in SeLEx, we start with an initial large library to probe diverse yet structured guide RNAs. And for this, we keep the ends of our library constant so that we can PCR amplify them between rounds. The middle of our library, however, is variable, and it's synthesized at each position with a mix of bases, where in this case, the wild type sequence formed 58% of the mixture, while 14% of each other base was divided evenly to create mutagenesis virtually in the synthesized pool. And here we can see the actual next generation sequencing of the pool we use for this study. And then putting this guide RNA pool through SeLEx steps, the process of SeLEx is the Sullenger lab focus, where the starting DNA library is transcribed into a RNA library, which is then added to Cas9 to form ribonucleoprotein complexes, which are then isolated via a selection step determined by the biology of the Cas9 ortholog in question, which we will discuss more in the next slide. The RNA was recovered and subjected to reverse transcription to cDNA, and then the cDNA was PCR amplified to reform and now enriched DNA library that can be transcribed once again to RNA. And the power of the process is that it is iterative, allowing us to start with extremely large libraries, as 1 × 10⁻⁴ and even 1 × 10⁻⁵, and isolate these down to hundreds or thousands of sequences. So Cas9 biology actually influenced this study a lot, as when we applied the SP-Cas9 method of directed evolution, we were not able to recover we were not able to recover a signal to noise ratio when evaluating the optimization of the assay. So we looked at a paper by Urek et al. And we saw that upon Cas9 cleavage of the DNA target, there is a rapid PAM distal release from the complex, and then a slower PAM proximal final dissociation. So we hypothesized that if we pull down the PAM proximal side, we could enrich for activity partially relative to this distal release. But this also gave us an idea of a potential negative selection where we could also pull out the PAM distal side of the complex to obtain enriched amounts of guides that had not released and potentially not cut this other side of the DNA. And a lot of this was carried out using radioactive refinement that helped us find a high signal to noise ratio after several iterations. So for this, we radio labeled the guide RNA, encompassed it with a Cas9 protein, to which we then added a biotinylated DNA target that was biotinylated at either the PAM distal or proximal side. And then upon pull down with stratavidin magnetic beads, and testing of different conditions at this point to wash the non-specific interactions, we were able to pull down using a magnet and measure an acetylation counter to evaluate the fractions of radioactivity that were bound to the intended beads as well as the washes. And then this allowed us to understand a signal to noise ratio developing over time. So here we can see the optimized signal to noise ratio after several optimization steps reached almost 26:1. So the pool noise was consisting of the guide RNA randomized, while the wild type signal was consisting of the guide RNA wild type sequence that we knew had cleaving function. And we can see that this was a significant signal to noise and suggested we could move on with our selection. After about 6 rounds of enrichment, we considered our selection done, as when evaluating our selection, we saw that while the no treatment retains the higher band, and then the positive control is cutting down this band of DNA, we evaluated the different pooled rounds at round 5 and round 6 for both a positive selection, which only pulled down pan-proximally, and a mixed selection that pulled down pan-proximally and distally depleted. So here we can see P5 and P6, we have increased cutting, and M5, we have already some cutting, and then M6, it is increased, suggesting that these selections were iteratively improving the capacity of these pooled guide RNA libraries to cut. So at this point, we sequenced this, and we observed that there was a high sequence diversity present with the wild type here at the top, and a consensus that is also close to the wild type, but cut off at the library 58%. The initial library compositions. And even then we can see that there's some bases that are steering away here from this consensus and from this initial library presence. And there was, for both positive and mixed selection, several mutations where we had between 7 and 32 mutations present in the top 50, which are depicted here. Furthermore, when we took these variants and we put them in vitro cleavage assays, we observed that these can outcleave the guide RNA in vitro. So when we have a piece of DNA and we carry this out in a test tube, we see that different guide RNAs. Okay, so we can see that the different guide RNA scaffolds, when applied to 4 different GFP targets, A, B, C, and D here, can cleave some of them better than the wild type. But we also can appreciate that, for example, M1 here can outcleave the wild type in this target D, but then in target C, it actually performs worse. So this suggests that the guide RNA, and this is a principle that I will be kind of going over a couple times, but it seems to be that a guide can be very specific to a specific target in terms of having a higher activity. So we wanted to test these next in cells, but first we performed a sequence logo of the in vitro cleaving variants. And we saw when arranging the sequence logo by the canonical secondary structure of the guide RNA, that a lot of the evolution of the bases had complementary bases on the other strand that were predicted to be bound that were actually evolving to be together. So we can see T's and A's here, AT, and over here also in the stem loop, G's and C's. But in general, this was suggestive to us that the evolution had a strong evolutionary drive coming from the secondary structure. And then at 5 different sites here with asterisks, we actually had a departure from the canonical sequence. So we went ahead and tested these different variants, first using plasmids by liposome transfection into cells. We waited 5 to 7 days for knockout and then evaluated these by flow cytometry. And we can see here at the bottom of our graph how knockout was actually improved with a lot of our different variants from both the positive and mixed selection. However, we wanted to also test this using ribonucleoprotein complex, which have become a leading method of transfection and have reduced off targets relative to plasmid delivery. And here we can see we essentially formed ribonucleoprotein complex and liposome coated them, transfected them, and also waited 5 to 7 days prior to flow cytometry measurement. And we can see that several of our variants actually were capable of outperforming the wild type scaffold. Here we can see, but these were fewer than the ones that we saw by plasmid transfection. However, the result was also significant. So we were wondering if we were to find cellular genomic targets rather than the current GFP targets that we had evaluated that were predicted to have poor folding if our guide RNA scaffolds would be able to rescue these. So we carried out a short bioinformatic pipeline and found a few different genomic targets in MBTPS2, KIF26B, and ERBB2, formerly HER2. And we saw that when we fold these, the wild type scaffold here at the top, let me get the arrow there. We see that there's disrupted repeat anti-repeats. And this was consistent for all 3 of them. But however, when we use blade 2 scaffolds, we see restored repeat anti-repeat regions. And this suggested to us in the minimum free energy that perhaps that we could have an improved function. When we took a look at the dot plots, we observed a similar trend coming from these, but at a more high throughput level where several more hundreds and even thousands of structures that form the overall folding landscape are evaluated. And upon testing, we observed that these guide RNA variants improved the gene editing at these genomic targets for all 3 targets significantly by up to 400%, the case of KIN26B. And this to us suggests that there is a strong relationship between secondary structure folding that the guide RNAs variants can actually rescue. When we evaluated the outcomes of editing by indel size distribution, we observed that these resembled wild type with deletions that were specific to each site that were targeted. So here we can see MBTPS2, for example, the -5 deletion is consistent, at least similar result between the guide 41 and also wild type. And while this is normalized to the total efficiency, so that's why they are the same, but the proportion of the actual indel distribution was not significantly disrupted between the 2. So when we take a look at these guide RNAs that we pulled out that had very specific and obvious secondary structure changes that we were able to restore, this made us think about, well, how could we potentially match the thousands of guide RNAs that we had, scaffolds that we had to new targets that did not have such obvious trends. So currently, the evaluation of CRISPR efficiency is very time consuming. And this made for us to having a library of over 100 trillion guides and then a final round diversity of about 30,000 of them. We were only able to test about 120 of them. So most guides remain untested. And this is a problem just widely across the field. So we wanted to find a way to test these guides more rapidly. So initially, we started by taking our single round pan-proximal selects and evaluating whether this could correlate strongly to cellular activity of RNPs. So we did a single round, we synthesized a library of 150 guides, and then we put them through one round of selects and then evaluated whether the enrichment after selects would correlate to RNP activity in cells, in HEC293 cells. And we saw that the correlation was weak. And you can see our square of 0.41. And we were wondering, well, since a lot of our mixed selection guides actually perform better, could we incorporate a PAM distal depletion to enhance this signal? So we incorporated this into a single round where we initially carried out a depletion, and then we added an enrichment part to this single round. And then we observed that there was an improvement in high confidence samples to an R square of 0.62 correlated to cellular guide RNA activity after RNP transfection. So this was encouraging, but we also wondered if we could even farther clean this up. And for this, we hypothesized that a mixed selection could be achieved by a gel shift, where, as you can see in this slide, there is several different bands, and they actually correspond when computed for the total size of the guide RNA Cas9 complex shift of the DNA band. We have all the different species, and we could actually isolate some of these bands and evaluate the cutting efficiency of the guide RNA. And when using this method, we actually found an even higher correlation, going up to 0.77 R square to cellular RNP activity. And this suggested to us that highly quantitative methods of in vitro cleaving can actually pull out a lot of trends that we will see also in cells and give us a characterization in vitro of the activity of our guide RNA scaffolds. So we wanted to test whether this could be done in multiplex. And for this, we combined our 150 scaffolds to 3 different mixes of targets. And we carried out essentially 3 separate spacer scaffold combinations of 3,000 and 3,100 for the last 2, divided by GC content, where there was a high, a mid, and a low GC content, including several targets that were close to clinically relevant sites. So here you guys can see the list. And when we carried this out, we observed that gel shift using this method of selects over a single round was actually found a strong correlation still to our quality control GIF-PQC depicted here. As you can see, R square value is above 0.7. But we also found that the predicted activity for a lot of different ones of these 150 scaffolds were actually capable of outperforming the wild type. So here in these violin plots, there's yellow dots. So these yellow dots correspond to the place where the wild type scaffold falls in activity range. So you can see that the wild type scaffold has varied levels of activity, but in some, especially in very low levels of activity, we see that the rescue can be quite significant. And here we can see this, for example, here with factor 12, X and 8, and we can see ERBB2 as well, which was actually here in the mix of high GC content. We can see ERBB2 goes up quite a bit, and this was actually one of the ones that we had tested previously. So suggesting that misfolded targets have a higher upside. However, all targets can benefit from using these alternative scaffolds that once you test several of them, you can actually find one that does better. And this can be done in high throughput. Also, this gel shift assay revealed to us some trends in our scaffolds where scaffolds that were evolved that had mutation numbers between 0 here in the far left is actually the wild type. But up to 20 mutations, we can see that there is significant activity from guides that have this number of mutations. But beyond this, we actually saw lower levels of predicted activity. And then at the bottom, we can also see when we compare scaffolds evolved via positive selections to scaffolds evolved via mixed selection, we saw that the mixed selection scaffolds, which had PAM distal depletion and proximal enrichment, actually performed overall, had more variants that performed better in our assay across several targets, suggesting that the need for negative selection is actually improves the outcome significantly and is able to match the wild type activity range. When taking a lot of these scaffolds, to be specific, 6,700 pairs that had high confidence reads, and converting them to secondary structures of using dot plots, and then comparing the base pair probability, separating these dot plots into individual base pair probabilities of each of the different residues to any other residue in the guide RNA scaffold, we found that there was several significant secondary structure patterns that emerged. Here you can see this in this dot plot heat map here on the right, where several positions were significant, but more salient than the significant positions is, as you can see, the red, blue, cool, warm map. In red, you can see positions where the proportion of the guides that actually follow these trends is very high, and approaching 0.8 to 1. And these actual positions that were highlighted were actually highly correlated to, were actually the same ones that have been involved in the canonical structure of the guide RNA. But we also observed some new emerging areas, like the top right, where we see this cloud of binding between the target and the scaffold that actually ends up being beneficial for cleaving. So this suggests that there are a lot of trends that can be elucidated using high throughput guide RNA testing. And when we took this data that we saw so structured and threw it into a deep learning data algorithm, we actually see that the ability for using a held out test within the data set, we see that there is a high ability to predict the guide RNA efficiency and selects activity using this deep learning prediction, suggesting that the data is highly structured and there are trends that can be rescued from this. And this encourages us as we this initial test of multiplex guide RNA evaluation is at initially testing 10,000 was actually a small display of what the numbers that we can scale by doing larger libraries, especially of the number of guides that we can test. And this brings me to some of the future directions and work that I've been doing at my postdoc lab, where I have been learning in the Chory lab about phage-assisted continuous evolution, where the evolutionary pressures change, but we are able to create both positive, also a negative selection by coupling the activity of guide RNAs to the production of phage through P3 presence in a circuit. And the process of phage-assisted continuous evolution has the potential of evolving also the protein, but we are also looking at the guide RNA, potentially evolving this farther. And the potential that we see a lot is related to the potential to bring in blade selects enriched guide RNA libraries, providing us thousands of starting points for phage-assisted continuous evolution, which has previously always used smaller libraries or evolution from an initial single point of wild type function and a wild type sequence. So this potentially gives us the potential to seed the sequence space and then access mutations that are more closely related and explore a deep sequence space with more precision. So this laboratory has a strong expertise in turboprams, which enables high throughput feedback driven pace automation using on-deck sterilization, automated media prep, evolution prep, tip storage, real-time sampling, and high throughput turbidostats, which allow them to explore in a rapid way several parameters and evolve potentially multiple different targets to on and off target pressures. And currently, this turbopram system, the robotic automated system has already accelerated our exploration of the Cas9 circuit, signal to noise refinement, where we currently have a system that we are going to use for directed evolution of the guide RNA using libraries from Select. So these will potentially provide us with outcomes that we do not, there's no precedent for, and will allow us to access new mutations that were not being able to be accessed previously. So in summary, guide RNAs have significant sequence diversity and can improve Cas9 efficiency via RNP and plasmid delivery. But also single round selects is highly correlated to RNP activity in cells, and selects libraries can provide thousands of functional library pooled starting points for PACE, which is something that we're going to explore and evaluate. But finally, blade selects has high data generation scale and lower associated costs that provide the optimal foundation for training sophisticated deep learning and generative language models. So I want to acknowledge the members of the Sullenger lab and Chory lab, as well as the Gorotkin lab that all collaborated to this. In the Sullenger lab, Corey Bush, who developed the first Cas9 evolution, Jonathan Zhou, who is working on a prime editing directed evolution, and Amos Yan, who has assisted throughout all of these different ones with a lot of expertise as well. And from the Gorotkin lab, Jan Gorotkin, who has been our computational kind of mentor and has helped us put together some of the evaluation of secondary structure alongside Julia Corsi and Ying Sung. And then Chory Lab, which is my postdoc lab currently, where I'm working with Chini Ma, who is an expert in mathematical modeling, but also the use of these automated systems. Ryan Beaulieu, who evaluates who's putting together the actual automated system, and Winston Kian, who will be joining us to analyze off-target sequences, as we are trying to also make this system also improve upon off-target effects going forward. Thank you. And yeah, I would be happy to take any questions.
Just a reminder to put your questions in the Q&A function, and Ronald will moderate the question period. But I just wanted to congratulate both of you on really cool work and really innovative ways of using the selection process to show kind of a new and different direction that we can take this evolution process and incorporate these aptamers, DNA enzymes, RNA enzymes, ribosome kind of situations. It was really cool.
Thank you, Aaron. Again, part of it grew out of frustration that Telmo mentioned that we were trying to edit different things. We're interested a lot in blood coagulation, so we were trying to edit specific mutations, say in different say factor 9 or factor 8 mutations in um, uh, but couldn't hit certain targets. So again, it was really frustrating to say, hey, we want to edit, not to go and inhibit factor 8 or 9, but we were trying to interested in repairing them. And then when we were trying to hit certain sequences, it became challenging where it was, basically virtually impossible, it seemed like, to make a guide that would hit a specific spot. So it kind of came out of, frustration for that. And also just thinking about it from, as you know, Aaron, whenever we put new sequences next to aptamers, you can screw them up more often than
Absolutely.
not, unfortunately. And so we started thinking at it from the aptamer perspective and say, hey, we're switching out this DNA, targeting the so-called spacer domain. We're kind of stuck with it often. And so if it makes a negative interaction with the scaffold slash aptamer like domain, then we're in trouble. So since we were stuck with the DNA targeting spacer domain, we said, well, can we switch the other piece of the equation? In the past, when we had the aptamer, we'd be playing with the sequence around it and doing
Yeah.
selection. They could accommodate the aptamer, whereas here we actually had to flip it on its head and say, can we change the scaffold at all to be able to accommodate new DNA target domains? And I would say the thing that was probably the most surprising to us initially was how much you could change it. It still had to
Yeah.
work. So we didn't know, would we get back only the wild type sequence when we did this initial approach? And it turns out you actually have thousands of variants that can maintain activity. So there's a lot more flexibility
Yeah. It's really cool.
than when we started. So, but it really grew out of frustration of trying to edit particular sequences where we couldn't. So,
I think that's most of the best solutions grow out of frustration in science for sure.
Yeah, and we can see that there's a strong possibility of being able to use these kinds of selection approaches and evolution approaches for any RNA guided endonuclease. There's thousands, hundreds of thousands, I believe, of CRISPR variants currently out
Hmm.
there in databases, and all of them are going to have a distribution of efficiencies, and any of them can be potentially evolved to have like guide RNAs, an army of guide RNAs to improve efficiency.
It's just so exciting to see like the worlds collide and see how they can be taken in an innovative direction for sure.
also, we felt like the the CRISPR world is so hyper competitive. We
Yeah,
said, if we're going to do something in the space, let's do something where we can kind of bring a particular expertise to. So we
exactly. Yeah.
thought we had so much experience of doing these type of screens. Although I have to admit, it took a long time to sort of set up the screens and figure it out because it's not just simple aptamer selection. Now you're selecting for this activity, and it's really a testament to both Corey Bush, who Telmo was showing here, and Telmo himself, that we had to really redevelop a way to do the selection, not just for binding, but for activity. And it turned out it was different for the different
Hmm.
cases, which again, and it turns out we're doing prime editing now is Telmo mentioned Jonathan Zhao in the lab, and it's a totally different, optimization of how to do the selection. So it,
Yeah.
as you know, from doing selection against one protein target to another, that
Yeah.
it's sort of customized. This is even customized in an additional way, not just for
Yeah, absolutely.
binding, but for activity. So that's been sort of an adventure, but actually, it's been lots of troubleshooting and learning lots about how you get out what you select for, as you know. And
Hmm.
so you have to be smart about how you set up your selection. So Telmo did a great job of this proximal distal observation of figuring out how do you deplete things that are probably binding but not cleaving. to
Yeah,
really get us to where we needed to go.
yeah, absolutely. Ronald, did you have questions? I see, just a reminder, you can put questions in the Q&A.
I always have questions. No, very nice and elegant work. So maybe a question to Telmo, because I think most of what you did is you designed the scaffolds mostly on one target. So do you think that the way that you introduce the study, that you optimize the study, can that introduce bias, kind of a bias in making it general?
Yeah, for sure. Definitely having selected to a GFP target means that these scaffolds will primarily, that we're repurposing towards other targets were evolved to a first one. And in a way, carrying out selections to a couple more targets, I think will answer, your question, I think, fully, where we will be able to see, GC content, are these better targets with specific GC content, or is it, primary sequence? And some of it we can see here um, when we evaluate them against different targets. Yeah, you can see that some targets can reach very high levels of activity, but other ones, even though we are improving the activity, they don't rise all the way, all the way to the top, basically, as you can see. Um, and this can, we will evaluate, I think, new targets and compare them also again by these like high throughput methods to see the different distributions that we're getting. But also, I think that's somewhere where, using like pace-based evolution, where you can have ongoing mutagenesis. It's starting out with these libraries towards new targets could possibly evolve a few more mutations that actually bring this up and adapt them to these new sequences.
So Ronald, can I comment on that? So I agree with you. I
Yeah,
always
definitely.
concerned that we pick a certain, again, to go back to us, we were talking with Aaron, a certain DNA targeting spacer sequence, and we're selecting in that context. So I think you are somewhat, biased and maybe even addicted to that sequence. And that if when we do it with a different sequence, you'd predict you'll get somewhat of a different set of answers, and there'll probably be some Venn diagram where some of them will overlap and some won't. And what we'd love to do is to do enough of those to develop machine learning where we could more computationally do it and predict, and so I think there's an element of having to do more of this and then use the information we get for training and computational. But I totally agree with you. If we started with a different, spacer domain, we're going to get a somewhat different answer, if not a totally different answer. And there's going to be somewhat, overlap and more or less. Now, one of my fun things to think about doing is because this is all in vitro, we can toggle theoretically the DNA targeting domain between rounds of selection. And so there's an idea that we've had is, can you develop a generalist that say can go, by switching in different DNA binding or targeting domains between the rounds. Now that's going to make it more complicated how you build the library and do the selection, but would love to think about doing things like that. So one of the things that's nice about this in vitro evolution, you have a lot of control of it, just like Telmo was showing, so you can do negative selection. We could also think about trying to build in tricks like that if we're smart enough to come up with them. Or again, can we, use selection to get rid of off targets, so we deplete the things that may hit an unwanted target as well. So I think, people out there, encourage them to think about these approaches and how to be creative about trying to address, and use them to address some of these issues. But totally agree, Ronald, this is sort of the tip of the iceberg, and that we have to be, utilize the tool to get at those type of issues.
So do you think that you need to repeat this trick or this fantastic strategy like multiple times, and then when you do this like 10, 20, 100, 1,000 times, and you keep on feeding this in the large language model or just AI, as I call it, do
Yeah.
you think that the predictions are getting better, better, and better?
Telmo, I'll let you answer
Definitely.
that first.
But yeah, definitely, I think that there's going to be a threshold at which like the actual learning of the model will plateau. Um, there is potentially uh, if we were to do individual multi-round selections, I think that that number would be higher because every single round we are kind of making the guide RNAs more addicted, as Bruce said, to the sequence. But I think if we do a multi-target evaluation, I think we can get enough data, even from our current data set of guide RNAs. If we just have enough targets and enough guide RNAs, I think we can get enough data for each of these data points to use for machine learning. Generative models is the goal. And I believe we have enough guide RNAs. If we take all 30,000, for example, and we combine them to, 1,000 targets, then we would have a very large number of training points. We kind of intend to start scaling these up just so we can get enough data in without having to do hundreds of selections multi-round.
Yeah, and a question I think in the same line is, so what I see in literature, I have the impression that there are new kind of Cas9 proteins are coming out like every day, right?
Yeah.
And this is only Cas9. Well, you have many other orthologs. So how does that impact your strategy?
Yeah, there's definitely a lot of Cas9 proteins. I think our strategy right now is kind of more towards there is, some Cas9s that have made it past a lot of evaluation for on-off targets and have become like very close to the clinic. SPCas9 specifically, it's the farthest along progress one. So I think that to generate therapeutics that can have like an agility of being evolved or being predicted for this Cas9 that is already in the clinic, I think that's an initial goal. Since there is obviously going to be a lot of other Cas9s, but they're going to until they reach this stage of preclinical and clinical development that SPCas9 has currently reached. I think that we're kind of focusing more on the battleground that we have currently to get things to be efficient and also safe for patient populations. And that's
So
kind of
I
like a
would
lot.
add,
Go ahead.
so I would say I would add to that, Ronald, actually, I actually think that those new Cas9s that are coming out, that this strategy can actually be really useful for studying them versus trying to optimize them for therapeutic application like Telmo is talking about. So, we are lucky in the sense that we knew a lot about SP-Cas9's guide RNA and the SA, but as you know, there's a new Cas every minute, like you're saying, is coming out. And so when you start to talk to people, they don't really know what's important in those guide RNAs. Now, I think what our data says for SP and SA, it's very consistent when you look at the crystal structure, it kind of makes sense. But for those, you don't really know what's important. So I think this is actually a way to interrogate from a basic, fundamental understanding perspective what's important in those guide RNAs, what structures are important. It sort of gives you phylogenetic covariation sort of information early on to think about, what base pairs are really important, like Telmo said. So I think it can be very useful, this approach, not just for optimizing, but for understanding. So for a different application of the technology, and that's really what, if you go back and even look at the first selects paper, that's how it was, why it was performed. It was really trying to understand the structure of a stem loop structure involved in, regulating T4 DNA polymerase, uh, regulating the translation of that protein. And so I think, it harkens back actually to the first early days of how selection was originally set up as really a mutagenesis system to look at, basically RNA structure function. So I think it can be very useful this approach for interrogating those new guide RNAs to understand them, short of having to do a crystal structure and stuff for all of them, to try to understand them. So I think it be both valuable for optimization, like Telmo was saying, but also from those basic fundamental type of studies.
Yeah, I agree on that. So and so what so what if we move fast forward to the future? So I think in the end you want to develop something for, I would say, for a clinical pipeline. So I think most of the study is like an in vitro cleavage assay, and how to move this to the preclinical animal models, and then maybe also the next step, how to move this into the clinic. So how can this be part, or I assume that this can be or should be part of a clinical development pipeline. So how to introduce this in a pipeline, and do you already have any evidence in any in vivo models?
We currently have just studied these in cells, like HEC293 cells, and that's kind of where we have some evidence that these do like also improve the activity. But I do agree that it is something that moving into animal models would just kind of prove that that this extends beyond cell lines. And we would think that based on the different work of chemically modified guides, which have shown that these also transition from through the different from cells to animals in a predictable way of improvement. I think this suggests that the guide RNAs should be able to do that, and it's something that I think we're looking at. Um, at the moment, we're more exploring, kind of figuring out more the complete, a more complete kind of picture of what guide RNAs we can have and how high the activity can be. Um, but I do think moving into animal models, and it's part of our plan for the future, especially also using the pace evolution system. Um, Part of that it will be, I think, validating, and I think a lot of our current also funding is focused on moving these beyond the in vitro and the basic cell line. Um, in evaluating also, there's a lot of mutations specifically that are patient mutations. Um, so getting into this as well as also, the base editor space, and as Bruce mentioned too, evaluating prime editors that are like all of these variants of CRISPR are more clinically relevant, than the basic CRISPR, but it's all kind of like interconnected, and you're trying to evolve slightly different functions to enhance the specific activities of like these new complex proteins that are being used in the clinic.
So I would totally echo Telmo here, Ronald, in that where we would like to go, sort of, and I mentioned it, we were frustrated about trying to edit factor 8 and 9 point mutations to restore activity in the liver because that's where those proteins are made. We're very interested now in getting base editors based on this approach or prime editing to repair those mutations back in the liver. So that's really where we're focused, and I think that's where we're going to have the biggest impact because again, there's lots of ways to knock out a gene, and sort of what we do is screen 10 things and one of them works pretty well to knock out something. But really where I think we'll see the translational impact is there. So we're trying to look back at the very things we couldn't do initially and apply this technology to it. Now we set it up asking the question, could you do this at all with GFP? But obviously the next step is to go after some of those targets that are tough to edit, that probably you have to hit a specific site to fix a certain mutation, um, to make it useful. So that's where we're headed. We aren't there yet, but obviously, we're thinking about, we can get stuff to the liver, we can edit things there. Whereas, we want to improve base editing and prime editing in particular, for liver targets. So that's where I think our focus will be going forward.
Yeah, and
Yeah.
a lot of like inborn neonate disease, liver diseases. As you guys have seen the case with baby KJ, it is currently a race that like you have to have a very large team, and you spend it's very, very expensive and also very time consuming. But it's also a race against time because the neonate inborn liver diseases deteriorate them so quickly. So we think that having a system where the optimization can be more automated, and even combining this selects and pace could potentially make a very agile system that can actually respond quickly, and given the new FDA guidelines on potentially umbrella coverage of different RNA formulations, keeping the protein constant, it suggests that there's room for creation of more agile systems of optimization that can deliver like guide RNAs that can edit ideally at these like sites that sometimes can be even patient N of one, but it's still are being attempted now.
And that's where I think AI is going to be really important, Raul, because we're looking at N1, that's the specific mutation. You gotta can use AI to help you design a base editor or a prime editor to go in and target that specific mutation. I mean, I think that's going to be hopefully this learning that we're doing as we do more of these will empower sort of AI to let us design those things without having to do a screen for every mutation to your earlier question. So, but again, I think that that's where we're going to see the particular value of this. And when it turns out when you go to prime editing, because you're switching not only a sequence on the 5' end of the scaffold, you're switching sequence on the 3' end, it's even worse problem, the folding. And so what we're seeing in prime editing, and if you look at the literature, the prime editing efficiency is even lower than it is for just, cleavage and gene inhibition. So we think, again, this approach will be particularly valuable there.
Yeah, no, fully agree on it. And I think especially for these liver disorders, I think you have a very limited time window. You need
Yep.
to develop this within what is it, 6 months.
Yep.
So the better you can do and the faster you can do the selection, the earlier you can start treating a patient. So I think it's very valuable, and I think this is very well understood. So moving, so we are already over time. So is it okay if we do two questions from the chat?
Of course.
Okay, so the first one from Adrian. First question, you mentioned that the guide RNA folding correlates with Cas9 activity. Do you think this mostly reflects overall RNA stability or specific structural features in the guide?
I think it reflects, a few different things and some things that we don't know also. But definitely guide stability will be one. Um, the complexing, I think, dynamics with the Cas9 and the complex, uh, like the functional complex output, I think it's quite an important one as well. I think potentially we'll be able to start answering these questions, doing potentially degradation studies of our RNPs. If we introduce something like a nuclease or an RNase, we would know if it's more related to the RNA stability or the potential complex. But I think answering a lot of these questions is important going forward, because then we will know kind of also how to combine this technology. If it's something that is related to stability, then potentially if we make these guys more stable with chemical modification at the tail or so, this could be optimal. But then it all depends on what answers we get when we start probing as to the why. And I think that's a great, insightful question. Thank you.
Yeah, I mean,
think that,
I think it's an important question. I don't know that we really know that we may be impacting both, but we selected on, for structure and binding to the protein. We could probably incorporate a stability side by like doing a transfection of libraries into cells and seeing which one lasts longer, but they're probably going to be interconnected. But we didn't intentionally select for stability, but we may be getting that as sort of a bystander by binding to the protein well. So, but it's a good point we need to look at more carefully. I agree with Telmo.
okay, very good suggestion. And then the second part of the question, um, or a second question. There's also evidence that shows that single guide RNAs and dual guide RNAs with the same spacer show different editing efficiencies. Do you think those differences are mainly due to the secondary structure or other aspects of the single versus the dual format?
Yeah, I think definitely the secondary structure is, I think, very important. Um, I would think that the differences are more related to this. But what was the second thing that they said? Related to secondary structure, or which one?
Or other.
Just or other aspects. So it's open. It's an open question.
I
Oh,
think it's, again, I agree with Telmo. I think it's likely. Obviously, it's going to be case by case basis to some extent, but I think in general, again, it's exactly why I think you see, differences if you just move down the GFP gene with different spacer sequences, you see differences from, different sites. So I think it's the same issue. It's probably largely secondary or tertiary structure, and the ability to form a functional complex. But again, it may be a case by case basis, but I do think it's one of the main drivers of the issue is the folding.
Okay. Well, then I think I will give the word back to Aaron.
Great. Well, what an excellent webinar to finish off 2025 with. We had a great conversation, really exciting kind of directions for the future. And so we just want to thank you again, Bruce and Telmo, for sharing your research, and for having such great discussion with it. And we hope to see everyone again in January in the new year.
thank you so much.
Thank you for giving us the opportunity and thank OTS.
Thank you
Yes,
so much.
thank you for being here. Thank you for
Yep.
this excellent presentation and discussion. Yes, and see you in Denver, I would say.
Yeah, see you in Denver, everybody.
That's right.